Today I'll be talking about the transmission electron microscope. The TEM was invented by Ernest Ruska in 1931, and at first its resolution was no better than any of the light microscopes that were around at the time, so it had little influence in the field. But slowly it was commercialized and the resolution improved to 2.4 nanometers in 1940 and 1 nanometer in 1945. And then further advancements um, were accomplished in 1990 to the aberration corrective system, the vacuum pumps, and the digital monitor and panels, which bring it to the powerful instrument that we know today. Because electrons behave by wave particle duality, we can tune the wavelength of electrons by the momentum. So by changing the voltage or how quickly we accelerate electrons, we can change how small of the wavelength of the electrons are, allowing us to view smaller nanometer scale materials. The wavelength of electrons happens to be about 10,000 times smaller than the wavelength of visible light, which again brings us to viewing nanometer and subatomic scales. The TEM is separated into three main systems. The illumination system, which starts with the electron source. There are two different types of electron sources. The first one being the thermionic emission source, where you can have a tungsten filament or a lanthanum boride or ceramic crystal that emits electrons by heating it up by passing a current through it. The electrons produced in the tungsten filament are not as coherent as the ceramic crystal, but the ceramic crystal requires a stronger vacuum to use. And then the tungsten tip, the field emission source of electrons, is the most coherent source that we know. It uses electron tunneling induced by an anode and another anode to accelerate the electrons to our source. And then there are several different types of lenses throughout our device that focus and defocus the electron beam. All of these are electromagnetic lenses where we have copper coils with currents passed through them to, again, focus or defocus the electron beam path or create a more coherent source. The first one that we have are the condenser lenses. The condenser lenses will spread or narrow the electron beam before reaching the sample, and it helps to eliminate high angle electrons. And then on the bottom right here, we see the objective lens. The objective lens is the most important lens in the device because it forms the first intermediate image of the sample. There's one place before and after the object or the sample. And then based on where you view the image, you can view the image along the optical axis or off to the side at a specific angle. And then there's also a point called the back focal length made by the objective lens, which is where you can view diffraction pattern images from the TEM. And then further, we have the intermediate lenses and the projector lenses later in the image formation and the magnification stages or systems. And these are mainly used to magnify the image that's produced on the fluorescent screen or on the monitor that we use to view. In the TEM, there are several electron scattering events that can occur. The first being that electrons are transmitted through the sample. This is your typical TEM image that you'll see. That can look something like this. And then we also have two different types of scattering. Elastically scattered electrons, where there is no energy loss from the scattering, can occur, and this will provide us diffraction pattern images at a specific Bragg's angle. So it gives us information such as the d-spacing or the crystallinity or strain of our particles. And then there is elastically scattered electrons or EELs imaging, which will tell you the energy loss that is found when the electron interacts with nuclei in the system. This can be used to probe band-to-band transitions, plasmon peaks, and phonon interactions in the sample. Then we have emitted X-rays that will be generated. This is called EDS or EDX. This can provide a compositional map of all of the elements that we have. Here we see that we're analyzing gold, nickel, and other elements in a nanoparticle. And then we can also produce Auger electrons and backscattered secondary electrons. There are two main imaging modes in the TEM, the first being the bright field on the left. Bright field images are viewed on the optical axis, so the electrons that are passed straight down through the image or the specimen. And this highlights the mass thickness of samples. So particles or a sample that has a higher mass thickness will be darker, where the background will be lighter. And then on the opposite side, on the right, we have the dark field images where the detector is placed off to the side at a specific Bragg's angle to collect the high angle scattered electrons. And this will provide information such as the crystallinity and despacing of particles better than the bright field would. The TEM can also be set up in what is called a scanning TEM mode. So the TEM traditionally will produce a parallel beam that will interact with the full sample and form an image all at once at the bottom. The scanning transmission electron microscope can have improved resolution because it will focus that to a point to then scan that point throughout the sample and create a map of the image. And this is a very powerful tool that is often used in a lot of research, so it's important to highlight. So some examples of where TEM is used. This is a nanoparticle paper where the motivation was to create high entropy intermetallic nanoparticles to use as a catalyst for semi-hydrogenation of alkynes in the oil industry. Their main question going into the study was to what extent is the surface of the nanoparticle ordered compared to the body and if it's homogeneous throughout it. In this experiment, they use in situ TEM, electron dispersive spectroscopy, and localized energy, electron energy loss spectroscopy. These are their as synthesized particles where they formed a bright image or bright field image on the left that can be used to size the nanoparticles, and then the EDS spectrums off to the right of that, which will confirm the amount of incorporation of all of the elements to ensure that there is a high entropy particle being formed. And then further on the right, we'll see more bright field images, but a diffraction pattern below them that is used to determine the crystal phase of the particles. Because another one of their big interests is the crystal phase and the disorder to order transition upon heating. And they found 3 different crystal phases in their synthesized particles. Then they go on to use an in situ TEM to heat up the particles and image it throughout the heating process. And they find that their synthesized particles will undergo a transition into more ordered intermetallic BCC particles and then into an alloyed or solid solution FCC nanoparticle based on the diffraction images that they can obtain. And then in another example of nanoparticles and the TEM being used, this study is motivated by improving synthetic capabilities and what nanoparticles we can create. They have a lot of applications in catalysis, like we just saw, energy storage, information storage, etc. And the main question was, can we form new metal mixtures that we haven't seen before in nanoparticles with a seeded nanoparticle synthesis? And here they use HADF STEM, which is a dark field scanning transmission electron microscope, and then STEM EDS. These are some of the particles that they analyze, again, showing some EDS maps to confirm the incorporation and the time throughout the reaction to show the change in incorporation. And at the bottom, you'll see different graphs doing the elemental percent of each of the elements. You can also do something called a radial profile of nanoparticles using EDS. And this will tell you how incorporated all of the particles throughout the 3D structure. So if there is a core shell architect, you'll be able to see this. In this top diagram here, you'll see that gold is more concentrated in the center of the particle, where the other elements are evenly dispersed throughout. So this indicates that the gold seed was used. And then this is the framework that they were able to create from all of the TEM images, where they used gold and platinum nanoparticles to create different architects and shapes and sizes of nanoparticles. So to summarize, the TEM has actually changed very little since its invention, but the resolution has improved significantly. And this has allowed for imaging of cells from microbiology, nanoparticles, and materials imaging. And because there are several scattering events that happened with electrons in a sample, we can get a lot of information from the TEM, such as elemental analysis, different types of images, energy loss, and diffraction images. And all in all, this has allowed the TEM to be as powerful as it is today and to really push the nanoparticle field in particular forward, with which we likely wouldn't know quite as much about.